Welcome to the respiratory zone where gas exchange happens. That's the real magic. This is Chapter 1, Part 3. So there are 3 common names that you should know, and we talked about these in Part 2, but they all mean a little bunch of alveoli. So that is lung parenchyma, acinus, and lobules. Yeah, these names all sound weird, but all they mean is a whole bunch, a whole sack, one little part of the alveoli. Those parts that look like a little bunch of grapes. So let's take a look at these parts. There are 3 different parts to the respiratory zone, and the first one is the respiratory bronchioles. Those are generation 20 to 23. They account for 3 to 5% of all the gas exchange. Then there's the alveolar ducts, generation 24 to 27. About 35% of the gas exchange happens in the alveolar ducts. Now together, that's about 40%. So the rest of the gas exchange happens in the alveoli, or the 28th generation. So a little bit about alveolar sacs. The alveolar sac is the 28th generation. The vast majority of blood and gas exchange happens there. There are about 2,000 little alveoli or sacs per lobule or acinus, 300 million in total in the adult lung. There are pores of Kohn and canals of Lambert, and we're going to talk about those in detail. The pores of Kohn and the canals of Lambert help the alveoli to stay puffed up. They provide communication between the alveoli and collateral ventilation. So here's a question for you. In an adult male, do you remember about how many alveoli are present in the lungs? We just talked about it. 300 million. Good job if you remembered. So in this particular part, the canals of Lambert are here and here. This is a terminal bronchial. This is an alveoli, one of these little air sacs up here. So here's the terminal bronchial, and here's one lobule, or acinus, or lung parenchyma. Now, if you notice, the canals of Lambert provide collateral ventilation between the terminal bronchial and then the alveolus to help those alveolus stay puffed up. There are 3 types of alveolar cells, type 1, 2, and 3. Type 1 is a simple squamous cell, and it makes up 95% of the alveolar surface. It's also known as a type 1 pneumocyte, and it's super thin. It's mainly so that we can diffuse carbon dioxide and gas. We can exchange them. The type 2 cells make surfactant. They're really important to us because without surfactant, we'd have a really high surface tension, and every breath would be like the first breath of life. It'd be so hard. We'd be so thin and working so hard to breathe. The other about type 2 cells are they have stem cell ability. They can change their type if need be. They have a higher metabolic rate than type 1 cells, and there are much less of them than the type 1 cells. The last type of cell is such as an alveolar macrophage or a phagocyte. Remember that phagocytes are cells that protect the body by eating harmful foreign particles, bacteria, and dead or dying cells. They're the garbage men of the alveolus, the alveolar macrophages, and there's just a few of these. What type of epithelium composes 95% of the alveolar surface? It's a type 1 pneumocyte or squamous epithelium. Which alveolar cells are the macrophages? Was it type 1, 2, or 3? Type 3, and there aren't as many of them. They're the garbage men of the lung. And which alveolar cells are considered to be the source of pulmonary surfactant? Type 2. Let's talk about the interstitium. In the interstitium, the alveolar capillary clusters are surrounded, supported, and shaped by the interstitium. It helps hold the alveolar together. The interstitium is a gel-like substance composed of hyaluronic acid molecules that are held to a web-like net of collagen fibers. The interstitium has two main components, the tight space and the loose space. So the tight space is the area between the alveolar epithelium and the endothelium of the pulmonary capillaries, the area where most gas exchange occurs. The loose space is on the outside. The loose space is primarily the area that surrounds the bronchioles, the respiratory bronchioles, the alveolar ducts, and the alveolar sacs. Lymphatic vessels, neural fibers are all found in this area, and the water content in the area can increase more than 30% in this area before a significant change in pressure happens. Kind of cool, right? Something you should know is that the collagen of the interstitium is believed to limit how far an alveolus can distend. Expansion of a lung unit beyond the limits of the interstitial collagen can actually occlude pulmonary capillaries and or damage the framework of the collagen fibers and in the end damage the wall of the alveoli. So what happens and how do they get distended? Well, they can get a blocked bronchiole and then air can be trapped. It can either be a partial block or a full block. And so what will happen is if it's a full block, atelectasis develops. That can be thick, sticky secretions. It can be inhaling a penny or a pea. So all kinds of things can cause a blocked bronchiole. The most common is just thick, sticky secretions. You can see a picture of a normal bronchiole and a picture of a blocked bronchiole and an area of collapsed lung in the picture. This is the end of part 3, gas exchange. Thank you for watching and you're ready for part 4.